To conclude this data science team production session, we'll have now Kenneth Longo. He's the Senior Director of Data Science at Wave Life Sciences. And he's going to talk to us about making data science accessible for domain experts and creating real-time value, business value. So Kenneth, go ahead.
Thank you very much, Cynthia. And I want to give a quick thank you to people from Knime as well as the summit organizers for the chance to have this discussion with you today. I'm going to go ahead and share my screen if that's okay. Move to my slides. So the title of my talk today is Data Science in Production, Making Data Science Accessible for Domain Experts and Creating Real Business Value. I'm going to jump right into the key problem and what I thought what I came to find the solution to be. So Wave, first of all, Wave Life Sciences is a drug discovery company that designs short DNA and RNA sequences to treat various life-threatening diseases. That's the quick background. We're a biotech company. We're a medium-sized company. We move quickly. And the question for us was, how can we quickly and reliably develop, test, deploy software, have all the modern tools of data science within that package, and do this all to answer key business questions. And the question that comes up in our setting often is, is there one tool or a tool that can give us everything from the backend data connectivity, UI, UX, and reporting solutions? And for us, that solution was the Knime analytics platform, and after that, server and WebPortal. This gives us access to robust data science tools, but importantly gives us a means for productionizing solutions for broad use and delivering those solutions via the browser. And I'm going to jump into some very specific examples of how we have used this at Wave. One of our larger pipelines is one that processes a particular type of data called qPCR data and performs some pretty sophisticated modeling on that data type. This workflow measures a biological molecule called mRNA or messenger RNA. It does this in a high throughput format on the samples that come in chunks of 384 because that's the design of the plate that is used to contain those samples. And what this workflow allows us to do is put together many different features, both inside and outside of Knime. Within Knime, we're developing and have developed components with interactive JavaScript-enabled graphics and tables that allow the users to perform guided metadata selection. We're able to extract important context properties from users, who they are, what they're doing, what they're working on, the machine they're on, etc. And we're able to come up with some pretty sophisticated, what I call control metadata schemes, where we define metadata and use that metadata as flow variables that ultimately define the prosecution of the workflow, but also define the protocol of what's being done in itself as the user advances through the workflow. We're able to add in some advanced statistical features using R, using linear mixed effect models, and some advanced graphics with ggplot2. And then there's the connectivity angle, where we're connecting to various databases and data sources and data APIs, some in AWS, some locally. And this workflow has allowed us to process many observations over the last 24 months. So the first step in this workflow is actually the scientist, after entering some metadata, performs a visual examination of the data. And this is actually, I think, one of the nice features of WebPortal, that people get to really interact as they move with the data, as they move through the workflow. And here, scientists are very used to looking at these different plate layouts, which are quite visual and usually presented as a heat map. And it's an early point in the analysis where scientists really get an intuitive sense of what's going on in their experiment. This could be a make or break moment, just looking at these heat maps and getting a visual sense of deformations in the plate, data outliers, systematic error that might occur in a row or column-wise error. And we interact with users at this point to often check things that have gone wrong, simply by them being able to share with us the link to the WebPortal job. The next step is another sort of even more interactive examination of the data, where the users are now exploring a scatter plot of some of these data points. They're able to select them interactively in the scatter plot view and see what those samples are down below in the linked JavaScript-enabled table. And this is really important for them to, again, generate more initial impressions of the data quality and what likely is behind the reason for certain outliers they may be seeing in their data. It may also give them an initial impression of whether their experiment has, quote unquote, worked. The next step after the user passes through that part of the workflow is some more advanced analytics modeling that includes robust linear mixed effect modeling, calculation of parameters. And the user is able to actually see within the web portal, the browser, some of these different static graphics that give them a quick visual readout of their experiment, but also some of the interactive JavaScript-enabled graphics. So here we have on the right what we call a B-score outlier heat map. So the user is able to actually interact with this and say, okay, this is a technical error, this is a biological error, and really get a better feel for their data. And finally, there's some additional modeling that goes on for particular types of data where there may be multiple doses. We call these dose response analyses. These are, again, carried out through the R interface and graphed with ggplot. We have some heuristics built into the model in this case that prevent users from trying to perform this analysis when in fact they may not have enough doses to justify using such a technique. So we're able to create basically using an if switch that prevents them from moving forward if in fact they don't have the requisite number of doses. And then they're able to review all the results in a tabular form, looking at the graphs and the parameters, their confidence intervals, goodness of fit stats. And the final step in this process is committing this to an AWS resident database. Another vignette here is a separate vignette relates to Chem Informatics. So our company is at its core a chemistry company. We design these molecules and develop these molecules. A big part of our company is medicinal chemistry and process chemistry. And one of the big things that Knime helps us out with is searching different chemical structures, doing what we call substructure searches, and arriving at simple lists of molecules that the scientists can then review. This becomes really super important because a lot of our chemistry is quite modular. We are designing DNA and RNA molecules, after all, and if you know anything about these molecules, they have a very typical structure. They have what we call sugars, bases, and phosphate linkers. And these fall into naturalized categories, and we actually use Knime increasingly to explore this modularity and help us understand our own chemical universe by using some of the features of the JavaScript nodes within Knime. Here's one showing what I like to call the baseball card view that we're able to arrive at after doing a substructure search. This has turned out to be a pretty important tool for interactions and collaborations between data science and chemistry groups at our company. And finally, I'd like to talk a little bit about bioinformatics. We do a lot of bioinformatics at our company. We do work in DNA and RNA sequences, after all. We like to know what relationships may exist between that sequence and other species, for example. And so we do a lot of bioinformatics that requires integrating command line tools that are commonly used in bioinformatics, tools such as Bowtie that produce long lists and tables of genes, alignment maps, species. These one-dimensional tables are not particularly useful, and so we've used Knime and the WebPortal interface to perform network analyses and to create these dimensionalized networks of gene, species, and off-target gene networks. And this has quickly become the lingua franca that we use within the company to assess whether or not our molecule is likely to hit its intended target, and to do that uniquely and not hit other targets that may exist within the genome or the transcriptome. And here we use edge weighting to delineate when there are very few mismatches indicating a strong link, you see a higher weighted edge here versus more mismatches, which are weaker links and a more lowly weighted edge. And with that, yes.
In that case, for example, if you have a strong match and you test it on rabbit, you don't want to, and it's very matched to a human, you don't want to test on a human if there was potential bad effects on the rabbit, correct?
Yeah, and in some cases, it may give us a very strong prior for what we could expect if we were to try that molecule in another species. Would we hit another gene, and can we predict based on the phenotypes associated with that gene what would happen? And so I'm not going to go into that level of depth. That's sort of the next level, and we're working actually with a lot of predictive and guided analytics to help us address those questions, and using Knime to do that as well. That gets a little bit deeper into the story, but it is fascinating, and that's exactly what you're suggesting is exactly what we use that for. Yeah.
Excellent. So the goal, is like you want to quickly prototype to make sure that you minimize risk. You quickly decide whether to build a drug or not, and the quicker you determine that, the more savings on time and money. Correct.
Exactly. And, in pharmaceutical development, we do sometimes test molecules in other species. And so it would be good to know if it has any chance of working in that species or not. That could also cause you to redirect resources.
Excellent. Well, Kenneth, really interesting topic. Sometimes it's a little bit on my head, but it's so amazing what you guys are doing and how you're quickly, using Knime to quickly determine or make decisions on these drugs. So I appreciate you being here and sharing the story.